So happy to be here. So I have nothing to disclose. So I kind of want to start off just by giving you maybe a little bit of background of what inspires me and, what brought me to do the work that I do today. So I actually was born in Alabama. I was born in Dothan, Alabama, a few hours away. There I spent a lot of time with my grandmothers. They're now in their late 80s and early 90s. My grandfathers, they actually passed away quite young. One, in fact, at the age of 39 from cardiovascular disease. So I think a lot about how my grandmothers were under immense stress, right? They grew up in rural Alabama during a time where, the systems of oppression were particularly heinous. But one thing that always stuck with me about my grandmothers is that they found something that they could kind of seemingly control. And for them, that was their diet and their lifestyle. They were really into healthy eating, really into exercising. And when I learned about the higher burden of chronic disease and cancer among the Black and African American population, when I got to college, I was really inspired to see how diet and lifestyle could be a way to reduce that burden. So as Dr. Judd mentioned, I wanted to give you just a brief snapshot of my research trajectory, specifically just to kind of make note of the diet score that I developed using the regards data, which was an excellent experience for my dissertation. So this score kind of characterized the aggregate contributions of whole foods and lifestyle exposures to systemic inflammation, which we know is associated with many different chronic diseases and with cancer. And though this won't be the focus of my talk, I just wanted to kind of make note of the score in case anyone wanted to talk about it. In the future, we've applied the score in many different contexts. My work focused mostly on colorectal cancer. So we found that the scores were positively associated with colorectal adenoma, which is the precursor to colorectal cancer, and with incident colorectal cancer. And the scores have been applied in other cohorts, such as the Iowa Women's Health Study, the NIH AARP cohort, all finding very interesting associations with cancer and other endpoints like mortality. So over the course of my master's and my PhD, there was this burgeoning of information characterizing the gut microbiome and implicating the microbiome in cancer, and also showing that the microbiome could be influenced by what we eat and by different lifestyle exposures. So I became really interested in understanding how diet and lifestyle might influence the gut microbiome and in turn influence risk for developing cancer and for cancer outcomes. So what is the microbiome? It's a really hot topic these days, but it does comprise of trillions and trillions of microorganisms that live both in and on our bodies. Some of these are similar across all humans and others may be special to you. And if these microbes are disrupted, it might promote risk for chronic diseases and other different types of outcomes. But I think one of the most exciting things about the microbiome is that it is potentially modifiable. So I focus a lot on the gut microbiome, where the majority of the microbes in our body live. These microbes are critical for helping us to absorb and process nutrients, and they also produce many bioactive products that have effects not just in the colon or in the GI tract, but also system-wide. So I think that the microbiome can be a great way to address kind of the cancer continuum. If we have a better understanding of the gut microbiome, we might be able to better prevent and diagnose cancer, improve patient prognoses, determine which patients may or may not respond to therapy, and ultimately help patients to go into remission and hopefully reduce their risk for recurrence. So kind of thinking ahead, long term, my overall framework of thinking when it comes to my gut microbiome research is to first understand how to conduct rigorous investigations of how the microbiome might be involved in development and progression of cancer and how it might influence treatment response. So we can better understand what are these high-risk microbiome characteristics. But then I think the next most important steps, perhaps, is to intervene to potentially change the gut microbiome from high risk to low risk to be able to better improve treatment for cancer or reduce risk for cancer. We can do this in a variety of ways: antibiotics, probiotics, all different types of medications. There's even more extreme methods like fecal transplants. But my method of choice, and I'll kind of allude to this throughout the presentation, is through diet. Because overall, I believe that eating a healthy diet not only improves your cancer risk profile, but improves your overall quality of life, right? So before I jump into how the gut microbiome may be associated with cancer and with diet, I first want to dive into how we study the gut microbiome. So in order to study the gut microbiome, you can collect a variety of specimens. Most commonly, you might get a tiny piece of biopsy from the colon, and that kind of tells you what bacteria are getting into those colon crypts. But more commonly, you'll collect a fecal sample in some way. And the way that you collect this fecal sample is really important. You want the collection method to preserve how the microbial signature originally looks. You want it to stabilize that microbial DNA. As often in field studies, participants are shipping in their fecal samples, sometimes at room temperatures or fluctuating temperatures. So you really want to make sure your collection method accounts for that. And it's also important that across epidemiologic studies of the microbiome that we're using common collection methods, because there can be some variation introduced by the collection method that you use. So if we want to develop these large, diverse data sources, it's important that we kind of field-wide come up with common methods of these. In our studies, we use multiple different collection methods to kind of account for this. So during my postdoc at NCI, I did a lot of work to assess what were the best collection methods for fecal samples. So for example, we had 15 volunteers provide a large stool sample that we split up into various tubes. So one of the tubes contained like 95% ethanol. We had FIT tubes and FOBT cards. Both of those are used for colorectal cancer screening. And then a tube with RNA later. We then took one tube within those collection methods, froze it immediately, and took another tube and left it out for 4 days to kind of simulate what would happen if someone was shipping their sample. We then used metagenomic sequencing, and I'll go over what that is shortly, to characterize the microbes in that sample. And we found that 95% ethanol did not do a great job of stabilizing the microbes, but all of the other collection methods did. We actually still collect 95% ethanol samples for metabolomics, if any of you are interested in that. So I'll come back to this later, but I want to make a note that these FOBT cards and FIT tests that are being used for colorectal cancer screening can be used potentially to establish a diverse prospective cohort. And I'll circle back around to that later in the presentation. So after we've collected our fecal samples, we then use sequencing to characterize the DNA in the sequence in the sample. There's broadly two different sequencing methods: 16S, RNA sequencing. So this is kind of hypothesis generating, where you're not really getting very granular detail. You can assign down to the genus level, and it's usually cheaper. Or you can do untargeted sequencing of all of the DNA in your sample. You can get really granular detail there. You can get species, what types of genes are present in the different microbes. And this is generally more expensive to do, but again, it provides you more detail on perhaps the functionality and other important characteristics that you might want to understand. After we've sequenced all the DNA in our samples, we put that into sequencers, and that tells us what sequences are present. We can bin those sequences into representative units that we can then assign to like taxonomy. So you may hear of fusobacteria or bacteroides. We can deduce that from different reference databases, and then we can calculate various microbiome metrics. And I'll go over a few of those just so you have a good understanding as I go through the results from some of my studies. So something that you'll hear of commonly is alpha diversity. So this is our within sample diversity. So for example, here, if we're thinking of these different shapes as different individuals, the person here on the left has two different shapes. So they would have an alpha diversity, or it's called richness of 2. And they would have a low evenness because there's 3 of one shape and only 1 of another. Whereas this person would have a higher alpha diversity because there's more shapes and a higher evenness. So richness can be characterized with observed sequence variance. The Shannon index kind of characterizes a combination of richness and evenness. We can even incorporate phylogeny, so like those little phylogenetic trees, and we can incorporate that information into alpha diversity metrics as well. So typically what you'll see is that a higher fecal alpha diversity is generally associated with lower disease risk or healthier states. I want to note that the same is not true necessarily for the oral microbiome or tissue microbiome. Beta diversity is our between sample diversity. So we're comparing person A to person B, for example. And you can see here the beta diversity here would be 2. Because there are 2 shapes in person B that are not present in person A. And again, there's a lot of different metrics that we use to characterize beta diversity, such as Bray-Curtis or Unifrac-based metrics, which again incorporates that phylogenetic tree into the diversity information. Once we have those different distances between 2 individuals, we can put together a matrix comparing each pair of individuals, and in more simple terms, just kind of project that information into a multidimensional space and create something like these principal coordinate plots where we're looking for clustering. So for example, healthy individuals who cluster away from diseased individuals. All right, so now getting into some of my work on the gut microbiome and cancer. These are just a few of my studies, and of course there are just an astoundingly growing number of researchers who are interested in the gut microbiome and cancer. So kind of a logical cancer site when you think of the gut microbiome is in the colon, of course, because of the proximity there. We know that colorectal cancer is a leading cause of cancer death in the US, and survival rates could use some improvement, especially for the later stages. Now, many of these individuals who have potentially curative surgery go on to have a recurrence, and that recurrence is often fatal. So what we really need is to identify these potentially modifiable biomarkers of poor colorectal cancer outcomes. So I have a few ongoing studies in Colocare. As I mentioned, we only did 16S sequencing in our pilot study, so we want to do that detailed shotgun metagenomic sequencing to ascertain more granular detail related to functionality. And we're also doing targeted sequencing of, for example, bile acid producing genes in these microbes. We're characterizing the tumor immune microenvironment to see how the gut microbiome might be associated with tumor immunity. We're developing dietary patterns that characterize or that are best associated with the gut microbiome. And the incidence of early onset colorectal cancers diagnosed below the age of 50 has gone up dramatically. So this is an area of focus for us, especially and also on addressing disparities. And finally, as I mentioned, yogurt was protective for survival. So I'm also developing a pilot intervention study where we can provide fermented foods like yogurt, kombucha, and kimchi to locally advanced rectal cancer patients to see how that improves the survival outcomes as well. So as I mentioned, you think colon, gut microbiome makes total sense, right? But I want to emphasize that the gut microbiome actually has system-wide effects. The gut microbiome is involved in regulating hormones, regulating the immune system. There's emerging evidence for its roles in metabolic function and insulin resistance and many, many other known and unknown pathways. So an active area of interest for me has been also investigating how the gut microbiome is associated with breast cancer. So when I was at my postdoc at NCI, I had the pleasure of working with investigators who conducted a study called the Ghana Breast Health Study. And they wanted to focus in sub-Saharan Africa, as they know that breast cancer incidence is dramatically increasing in that area. And kind of as a comparison point, we know that African ancestry women are more likely to be diagnosed at younger ages, later stages, and also with more aggressive subtypes of breast cancer. So in this study, women presented to hospitals in 2 large cities in Ghana, and they had biopsies for their breast lumps. And then we selected population-based controls from the areas that gave rise to the cases. And all of these individuals provided stool samples, and we used, again, that broader 16S RNA sequencing approach to characterize the bacteria in the stool samples. So we found some really strong associations of alpha diversity with breast cancer. So just to kind of orient you, we divided, these are tertiles of alpha diversity based on the control distribution, and we use logistic regression to calculate odds ratios. And overall, we found a 66% lower odds of breast cancer comparing those in the highest tertile of the Shannon index compared to the lowest tertile of the Shannon index. And you can see that the associations are even stronger for richness and when we are incorporating that phylogenetic tree. And intriguingly, we did find kind of similar associations across hormone receptor status and across different grades. And in the study, they actually had non-malignant breast disease cases as well. And the associations were strikingly similar for those women as well. So even though, the study was kind of cross-sectional in nature, which is of course concerning, and we want to see more prospective studies of the gut microbiome, it is, somewhat promising that we're seeing the strong associations across grade and for potential precursors of breast cancer. So again, here you see in the red and green, breast cancer and non-malignant breast disease cases, they kind of clustered together separately away from controls. And these findings were statistically significant after adjusting for many different confounders. And for our taxonomic associations, here, just to orient you again, I'm presenting relative abundance data where we're kind of dividing the counts of one taxa divided by the sum of all counts in an entire sample. And we found for every 1% increase in relative abundance of Bacteroides, there was a 5% higher odds of breast cancer. So pretty strong associations. And again, there's a lot of biological plausibility here. Some of these taxa may encode enzymes that are involved in estrogen deconjugation, contributing to estrogen burden, which we know is a risk factor for many subtypes of breast cancer. And again, they may also be involved in producing these bioactive microbial metabolites. And I'll kind of come back to some of these metabolites later on as a way to kind of orthogonally study the gut microbiome. But there seems to be a lot of biological plausibility and strong support for associations of the gut microbiome with breast cancer. So kind of as next steps, I'm a co-investigator on a biobank where we are creating a stool repository among women who are undergoing routine mammography screening in Florida. So in that study, breast density will actually be our endpoint. So that will really be interesting in terms of being able to investigate associations of the microbiome with the risk factor, an established risk factor for breast cancer. In terms of outcomes, we're investigating whether the microbiome modifies how breast cancer patients respond to therapy. And we're also investigating the role of the microbiome in disparities, as that is a major focus of mine. So kind of switching gears to that point, we know that breast and colorectal cancer, they're very disparate cancers. So even though Black women are less likely to be diagnosed with breast cancer, they're actually more likely to pass away from it and also more likely to be diagnosed with more aggressive subtypes of breast cancer. But even for the more treatable breast cancers, Black women are still more likely to die from those treatable breast cancers as well. For colorectal cancer, although we see that the incidence of colorectal cancer is largely decreasing, we still see these substantial racial gaps where Black Americans are about 20% more likely to be diagnosed with colorectal cancer and about 40% more likely to pass away from it. And even though, again, we're seeing these declining trends, the trends among individuals younger than 50 are actually increasing. People are getting colorectal cancer younger and younger, which is really concerning. And you can even think of Chadwick Boseman, who passed away in recent years at the age of 43 from colorectal cancer. So why use the gut microbiome to potentially study disparities? Well, first, I want to convey that, I'm sure you all know that race, is a social construct, and, that it's not rooted in biology, but, really justification for discrimination based on the way someone looks. Ethnicity, there's issues with the way we group group ethnicities together, group very heterogeneous individuals together into one ethnic group. But there are many exposures that are associated with someone's race and ethnicity, right? Like racism, psychosocial factors, differences in diet and lifestyle. But these exposures are really complex and oftentimes very difficult to capture in our typical research studies. But we do know that these exposures influence biology, right? So they may influence the gut microbiome. And for the gut microbiome specifically, we know that it is associated with many disparate conditions in the United States. And there's been a few studies that have demonstrated differences in the microbiome across racial and ethnic groups. In the Southern Community Cohort, they characterized the oral microbiome and compared differences between African Americans and European Americans, and found that African Americans had higher oral alpha diversity and differences in overall microbiome composition and abundance of certain taxa. Kind of going back to, again, my long-term plan, I think we always have to focus on those targetable exposures, and they didn't really have the detailed information on tooth loss or periodontal disease. And we know that periodontal disease is more prevalent among African Americans and is also associated with many disparate conditions. So that's a potential point of intervention there. So again, when we're doing these studies, I think an emphasis should always be those targetable or potentially targetable exposures. So in Amsterdam, Netherlands, they compared the fecal microbiome across various ethnic groups, again, finding strong differences in alpha and beta diversity. And sorry, just reminding you what beta diversity is here, but very strong differences in beta diversity and in certain microbes. And I really liked in this study that they tried to kind of adjust out these ethnicity related factors, but the differences still remain. So there does seem to be some accumulating evidence supporting that the gut microbiome may look different across racial and ethnic groups. So in the long term, one of my career goals is to characterize the gut microbiome across large and diverse racial and ethnic, or large and diverse populations, including a diversity of racial and ethnic groups and geographic locations. To support this work, I can leverage, again, my postdoctoral work, where we found that FIT tests and FOBT cards were acceptable collection methods to study the fecal microbiome, opening up opportunities to establish diverse prospective fecal microbiome cohorts in populations that are being screened using these tests. So the FIT test is a test for blood in the stool to screen for colorectal cancer or precursors to colorectal cancer, and it's often provided to patients in the setting sometimes they may have access to colonoscopy, but oftentimes they do not have access to colonoscopies who are in the screening ages of 45 to 75. And normally after these samples are tested, they're just thrown out, but we can collect these tests and study the microbiome. So I received an NCI diversity supplement nested within a larger colorectal cancer screening intervention to establish a biobank of these discarded FIT tests from diverse participants in the clinical screening population in Florida. And I hope to eventually take this nationally. This is a really unique cohort because it includes a very diverse understudied population. We have almost 50 participants to date, and almost around 80% of them are Spanish speaking. We have a lot of rural area residents, migrant farm workers, and many African Americans as well. So it really targets a population that we often aren't seeing in primary care or other clinics. So in the study, we are collecting very detailed information on potentially modifiable exposures like diet, medication use, physical activity, and also social determinants of health. So sleep, neighborhood, stress. So we're collecting all of that information to investigate how it might influence the gut microbiome. And eventually we hope to follow these participants over time to see whether or not they develop cancer, so we can understand how these exposures influence the microbiome, which in turn influence someone's risk for developing cancer. And I also, want to emphasize that a major limitation in the microbiome field to be able to understand how the microbiome is associated with risk is that all that we have right now is cross-sectional studies. The microbiome and cancer is kind of measured at one point in time. So, which is driving which. But if we are able to establish resources such as this one that are prospective in nature, we can really dig into cancer etiology. So lastly, I want to mention microbial metabolites, which I think is an interesting and creative way to kind of get around the lack of fecal samples that are available right now in prospective cohorts. As I just mentioned, most existing fecal microbiome studies are cross-sectional, and also many of them lack diversity. Ideally, what we would want is to have multiple fecal samples collected at multiple points over time, but we don't have that in many of the existing cohorts. We usually have blood samples collected at 1 or 2 points in time. So what we can do is measure these microbial metabolites, or the bioactive products that the gut microbiome is producing in the blood, to kind of get at the associations of the microbiome with cancer. And we did this in the PLCO, the prostate, lung, colorectal, and ovarian cancer screening trial cohort. We measured TMAO and its precursors, choline and betaine. TMAO is produced or indirectly by the gut bacteria, and also has been substantially associated with cardiovascular disease. But for example, we found a 90% higher risk of distal colon cancer, comparing those in the 90th versus 10th percentile of TMAO. So kind of my microbial metabolite work, it follows a similar framework where we are still investigating, the associations of the microbiome through these metabolites with cancer, and then ultimately hoping intervening to change the microbial metabolites themselves, such as through diet. So as I mentioned, we know diet influences the gut microbiome, and in turn, the gut microbiome produces these microbial metabolites, one such as bile acids, which is produced by bacteria, secondary bile acids are produced by bacteria and have been strongly implicated in carcinogenesis. So we conducted 2 studies. But first, we investigated associations of fiber, coffee, fat, and alcohol intake with circulating bile acids measured in the blood in 2 large study populations. We found that fiber and coffee were strongly inversely associated with bile acids that have been strongly associated with cancer. We also found that alcohol and certain fats like trans fats were positively associated with bile acids. We aim to replicate our findings in a cohort of US men and women, finding that fiber was similarly strongly inversely associated with cancer-associated bile acids. We wanted to replicate our findings in the setting of an intervention study. So I leveraged data from the polyp prevention trial, which is a 4-year trial of high fiber, high fruit and vegetable, low fat diet. The original aims were to investigate the effects of this diet on adenoma recurrence as compared to a regular diet. We measured circulating bile acids at baseline, year 2, and year 3. Investigated the changes in bile acids over time. We did not actually find that the dietary intervention affected the bile acids, but we did find that at baseline, when they measured longer term fiber via food frequency questionnaires, that diet was strongly inversely associated with levels of bile acids. So, there does seem to be some consistency with the fiber associations. There's a lot of biological plausibility as being that fiber actually binds bile acids to promote their excretion via the stool. But of course, we need to continue to replicate these findings and to conduct perhaps more interventional studies to continue to validate these findings. So, I hope I've given you kind of a good snapshot into my work. I, again, focus on diet and lifestyle, the gut microbiome, and have ultimate goals to figure out ways that we can reduce the cancer burden, particularly among populations that are disproportionately affected. To do this, I am actively developing microbiome studies in diverse populations. I'm investigating the associations of the microbiome with cancer kind of across the cancer continuum, from risk to outcomes. And I'm also hoping to identify targetable exposures that are driving cancer burden and driving cancer disparities, such as diet. So I would not be able to do any of this work without my amazing team. And here's my email and Twitter handle. So feel free to reach out to me if you like with any questions. And thank you so much.